interest in pluriBead, the cell separation method from pluriSelect. Your advantages with pluriBead: you can easily separate cells in only 30 minutes. And you can use cells from different mixtures of substances. For example, from the familiar buffy coat, from cell cultures, and even from untreated whole blood. This is how the method works. Catcher particles and sample material are mixed with each other. The target cells bind together with the catcher particles. The sample material then flows through a sieve. The particle-bonded target cells remain caught in the sieve. Finally, the target cells are separated from the catcher particles so that the pure cells are available for subsequent analyses. To start, remove the mixing container and the vial with the catcher particles from the kit. You can choose different volumes, for example as shown here in the image, 0.5 milliliters, 5 milliliters or 15 milliliters. Then use a pipette to add sufficient particles for the current sample to the mixing container. The sample material is then added. Now mix the container using your mixing system or using the pluriPlex universal mixer from pluriSelect. During the mixing, the required target cells bond onto the catcher particles. The separation system with the sieve is now placed on an empty centrifuge tube. And this separation system is in turn placed onto the mixing container. Everything is turned upside down so that the sample material can flow through the sieve. The target cells, which have bonded with the catcher particles, are held back by the sieve. Unbonded sample material flows through the sieve into the centrifuge tube. Both the mixing container and the sieve are then rinsed with a washing buffer. The sieved cells, including catcher particles, are still caught in the sieve. A connector ring is therefore pushed onto an empty reaction tube, and then the sieve with the target cells are placed on it. A special detachment buffer is added. 10 minutes later, the buffer is carefully pipetted up and down so that the cells are released from the catcher particles. If the valve on the connector ring is then opened, the released cells flow into the reaction tube. They are now pure and available for further experiments. With pluriBead, you can separate cells in only 30 minutes, even from buffy coat and untreated whole blood. It is easy to use. Furthermore, no centrifugation and no sample preparation are necessary. You obtain highly vital cells. pluriBead from pluriSelect.